Usually when we talk about a medical diagnosis, there's this expectation of clinical precision. Like, it feels kind of like engineering. You break your arm, the x-ray shows the jagged white line, and the doctor just points to the screen and says, well, there's the problem.
Yeah, exactly. It's very binary, broken or not broken.
Yeah.
And, that visibility is comforting because it gives us a really clear enemy to fix.
Right. But then you step into the world of neurodegenerative diseases, specifically Alzheimer's, and suddenly that X-ray machine feels, completely useless. We're looking at a diagnostic landscape that is incredibly murky.
Oh, absolutely.
Because we often think of Alzheimer's disease as just this problem of dying neurons, like a tragic accumulation of misfolded proteins that are slowly shutting down the mind. But for you listening right now, what if it's actually an epic, decades-long battlefield story involving a massive invasion by your own immune system?
And that is exactly the premise we are exploring today. We're looking at some truly fascinating research published in Nature Communications in 2026. And it's titled, Type I Interferon Drives Key Cell Responses to Amyloid-beta in the Central Nervous System.
Which sounds very technical, but...
It does, but it fundamentally shifts how we view this disease. Yeah. It reframes Alzheimer's from a simple, plumbing issue of protein buildup into an incredibly complex, evolving immune war.
Okay, let's unpack this. Because to really grasp the magnitude of this shift, we have to look at what is physically happening in the brain. So Alzheimer's and a related condition called cerebral amyloid angiopathy, they're characterized by amyloid-beta plaques. These are basically toxic clumps of protein.
Right, and those clumps build up not just in the brain tissue itself, but also around the blood vessels. And for decades, literally decades, when scientists looked at the immune response to these plaques, they focused almost exclusively on microglia.
Right, the microglia.
Yeah, microglia are the brain's resident immune cells. You can think of them as the local neighborhood watch.
But this paper takes a completely different angle, right? Because it's looking at the adaptive immune system, specifically T-cells. And that immediately makes me pause.
Why is that?
Because for the longest time, I was taught that the brain is an immunologically privileged organ. Like, it was supposed to be a VIP club behind the blood-brain barrier where circulating immune cells from the rest of the body just simply weren't allowed in.
Yeah, that was the prevailing dogma for a very long time. The blood-brain barrier was thought to be an impenetrable wall, but emerging evidence shows that barrier becomes highly dysfunctional as Alzheimer's progresses. So the researchers behind this paper, they really wanted to know what happens when those circulating T-cells actually cross the barrier and enter the brain.
Right.
And to study this, they needed a very clean model. So they used this specific genetically modified mouse called the APP23TG mouse.
Okay, wait, what makes that specific mouse model so useful for this kind of deep dive?
Well, these mice carry something called the Swedish mutation. So their genetics are specifically tweaked, so they massively overproduce amyloid-beta peptides.
Oh, I see.
Yeah, it mimics a progressive amyloidosis. This gives researchers a really clear view of the plaque buildup without other confounding factors, like neurofibrillary tangles, sort of muddying the waters.
So they basically have a perfectly clean window to watch the disease progress.
Exactly.
And they looked at two distinct time frames, right? First, an early disease stage when the mice were around 12 to 14 months old and the plaques were just starting to form. And then they looked at a late disease stage at 20 to 24 months. But how do you even know a mouse is experiencing the equivalent of late-stage cognitive decline? It's not like you can give them a standard memory test.
No, you can't. So you have to observe their innate behaviors. In this case, the researchers used a nesting task.
A nesting task?
Yeah. Mice naturally love to build structured, complex nests out of materials you provide to them. It requires motor coordination, spatial awareness, memory, all of that. But by 24 months, these genetically modified mice showed a severe decline in their nesting scores.
Wow. So what do they do instead?
They would just leave the material scattered randomly around the cage. It's a really stark behavioral indicator of the underlying neurological deficits that are being caused by the plaque accumulation.
Okay, so we have this really clear timeline, right? From early plaque formation to severe cognitive decline. And watching the immune response evolve over that timeline reveals the first major revelation of the paper, because the researchers used a technique called single-cell RNA sequencing to watch this happen.
Yes, an incredibly powerful tool.
I always find this technique mind-blowing. Like, instead of just grinding up a piece of tissue like a smoothie and getting an average reading of what the immune cells are doing, they are isolating thousands of individual cells and reading the exact RNA transcripts inside each one.
Right. It gives you unprecedented resolution. You can see exactly which genes are turned on or off in a single macrophage versus a single T cell. And what they saw was a dramatic shift in the landscape.
Right.
In the early disease stage, the immune response was entirely dominated by those local microglia. They were clustered around the plaques, basically attempting to eat up the amyloid-beta.
But in the late disease stage, the environment completely flipped. There was this massive influx of CD8 positive T-cells just flooding into the brain.
Flooding in, yeah.
To use an analogy for you listening, it's like having a water leak in a massive office building. At first, the local security guards, the microglia, they rush over with mops. They're trying to contain it. But the leak just keeps going year after year. And eventually, the local guards get completely overwhelmed. Their mops are saturated. And suddenly, the National Guard is called in, just swarming the building.
That analogy captures the scale of the infiltration perfectly. But this massive arrival of CD8 positive T cells, which are typically cytotoxic, meaning they are explicitly designed to kill infected or damaged cells, it raises a really heavily debated question in the field. Are these T cells actually helping to clear the plaques, or is their presence causing more collateral damage to the brain?
Yeah, that's my immediate reaction. Like, if the local guards couldn't fix it, maybe heavily armed military personnel just make a mess of the lobby. What does the historical data say about this?
Well, what's fascinating here is that it's completely split. Some past studies found that if you deplete T cells in animal models, the amyloid deposition actually accelerates and the cognitive impairment gets worse.
Oh, wow.
Yeah, which suggests the T cells are restraining the disease.
Right.
Like they are the good guys.
Okay, but I'm guessing there's a counter-argument to that.
A very strong one. Other studies found a direct correlation between the number of infiltrating T cells and the extent of neuronal loss. In some of those models, completely depleting the T cells halted the neurodegeneration entirely.
Meaning they're the bad guys. God, that is incredibly confusing for anyone trying to develop a treatment. If you boost the immune system, you might accelerate brain damage. But if you suppress it, you might let the plaques just grow wild.
Which is exactly why we can't just broadly label them as good or bad. We have to decode exactly why they're crossing the blood-brain barrier in the first place and, what their specific molecular marching orders actually are.
Right. So let's do a quick recap here to make sure we're all on the same page. Early Alzheimer's features local microglia trying to contain the damage, but late Alzheimer's features a massive invasion of CD8 positive T cells. So, to you listening, if you were designing a drug for Alzheimer's right now based on just this, would you try to boost these infiltrating T cells or block them? Keep that question in mind as we go deeper.
It's the central tension of the whole field right now.
So let's talk about how they actually get there. If these T cells are swarming a massive organ like the brain, they can't just be wandering aimlessly, right? The paper shows these beautifully stained images where the T cells are tightly clustered right around the plaques deep in the brain tissue. They basically bypass everything else to get to these specific toxic clumps. How are they homing in on them?
They follow a very specific genetic and chemical breadcrumb trail. The researchers discovered a unique subset of these CD8 positive T cells that were highly expressing what are called interferon-stimulated genes, or ISGs.
Now, wait a minute. Interferon is usually something that comes up in conversations about viral infections, right? Like, it's a biochemical alarm bell that cells ring when they detect a virus. Why is it firing off in Alzheimer's?
Yes, that's a great question. Type I interferons are a massive generalized alarm system for severe cellular stress, not just viruses.
Okay.
In the early stages of the disease, the microglia are the ones expressing these ISGs. They're sounding the alarm. But the researchers found a very clear baton pass. In the late stages, those microglia quiet down, and this newly discovered subset of ISG-expressing T cells actually takes over as the main driver of the interferon response.
So a specific unit of the National Guard sets up a command post near the plaque and just, takes over the alarm system. But how does that localized alarm actually summon all the other T cells that are still circulating out in the blood?
Through a beautifully specific chemical flare. These command post T cells secrete a chemokine called CXCL10. You can think of a chemokine as a biological scent trail. Meanwhile, the other circulating effector T cells, the foot soldiers out in the blood, they possess a specific receptor on their surface called CXCR3.
Like an antenna.
Or a very highly tuned nose. The CXCR3 receptor is specifically looking for the scent of CXCL10. When it detects that signal, the circulating T cells undergo chemotaxis.
Chemotaxis?
Right. They migrate straight toward the highest concentration of the signal, which leads them directly through the blood-brain barrier and right to the plaque.
See, here's where it gets really interesting. The researchers didn't just hypothesize this scent trail. They proved it using an incredibly elegant in vitro experiment called a transwell migration assay.
Yeah, this is brilliant.
Imagine a microscopic chamber split into a top half and a bottom half, and they're separated by a porous membrane, kind of like a tiny sieve.
Right, and the pores are just large enough for a single immune cell to squeeze through, but only if it is actively trying to migrate.
Exactly. So they put circulating T-cells in the top chamber, and in the bottom chamber, they put that CXCL10 chemical, or they put actual ISG-induced T-cells that were actively producing the chemical. Normally, the T-cells in the top smell the signal and squeeze down through the sieve, but the researchers used CRISPR gene editing to knock out the CXCR3 receptor in a batch of T-cells.
They essentially surgically removed the T-cells' noses.
Right, and when they put those blinded T-cells in the top chamber, absolutely nothing happened. Even with a massive amount of the CXCL10 signal screaming at them from below, without that specific receptor, they just sat there. It proves that this massive brain infiltration isn't just a random, chaotic, inflammatory spillover. It's a highly coordinated, target-specific homing mechanism.
Absolutely. It firmly establishes the CXCL10-CXCR3 axis as a mechanistically vital driver of neuroinflammation in late-stage amyloidosis.
Which brings up a strategic question. Let's do another quick recap for the listener. ISG CD8 positive T-cells sit near the plaques and release CXCL10. That's the signal. Other T-cells with the CXCR3 receptor smell the signal and swarm the area. So think about a time you followed the smell of a bakery to find a specific store. Your nose was the CXCR3 receptor, and the scent of bread was the CXCL10. If we want to stop the swarm in the brain, do we plug the nose, or do we eliminate the scent?
Both are theoretically viable targets for drugs. But before we talk about clinical therapies, we really need to look at what happens when these foot soldiers actually arrive at the plaque. Because they don't just sit there peacefully. They arrive, and they undergo a process called clonal expansion.
What does that actually look like at the cellular level?
Well, the researchers used another advanced technique, sequencing the T-cell receptor, or TCR. Every T-cell has a unique receptor, functioning almost like a fingerprint, and it's designed to recognize one specific molecular threat. When a T-cell finds its specific target, it gets activated, it realizes it has found the enemy, so it starts cloning itself, multiplying rapidly to build an army custom-built for that exact threat.
It's like a locksmith trying thousands of keys, finding the one that finally turns the lock, and then frantically cutting 1,000 copies of that exact key.
That's a perfect way to picture it. And in these late-stage mice, there was massive clonal expansion right in the plaque neighborhood, and these cloned T-cells were heavily armed. They were upregulating genes like GRASMA and PRF1, which produce granzymes and perforin.
Granzymes and perforin, what do those do?
These are proteins that are literally designed to punch holes in target cells and destroy them.
Wow. But here is the tragic irony in all of this data. They arrive, they clone themselves, they pull out their weapons. They prime their weapons. But they are fighting an uncleared, practically indestructible protein rock. The amyloid-beta plaque doesn't just die and clear out like a normal virus-infected cell would. The T-cells are attacking a fortress that just won't fall.
And that chronic nature of the plaque leads to a severe state of T-cell exhaustion.
Exhaustion.
Yeah. It's a phenomenon we actually know very well from chronic viral infections and oncology. The T-cells are hyperactivated for so long, constantly trying to destroy an indestructible target, that their internal programming forces them to put the brakes on themselves.
Oh, because if they didn't, what would happen?
If they didn't, their continuous release of cytotoxic chemicals, those perforins and granzymes, would basically destroy all the healthy surrounding brain tissue.
So they exhaust themselves on purpose to prevent a total meltdown. How is that burnout reflected in their genetics?
The highly expanded clones showed a massive cumulative exhaustion score. They strongly upregulated genes like TOX, LAG3, and most notably, PDCD1.
Wait, PDCD1, that's the gene that creates the PD-1 protein, right? Because anyone familiar with modern cancer treatments has probably heard of PD-1. It's an immune checkpoint. Tumors frequently exploit the PD-1 pathway to basically put attacking T-cells to sleep.
Exactly. It acts as a profound inhibitory receptor. When the T-cells upregulate PD-1, they're basically waving a white flag, signaling that they are too exhausted to continue an effective fight. And facially, the researchers mapped this beautifully. The exhausted PD-1 expressing T-cells were localized directly at the outer edge of the plaques.
Now, I have to play the skeptic for a moment here. Up to this point, we've been talking entirely about mice, the APP23TD mice. And, we all know the old joke in medical science. We have cured Alzheimer's in mice 1,000 times over. It rarely translates perfectly to human neurology.
That is a very fair skepticism. And frankly, it's exactly why the researchers didn't stop with the mouse model. If we connect this to the bigger picture, they obtained post-mortem human brain tissue cores from patients who had Alzheimer's disease to see if this same battlefield existed in humans.
But how do you map this kind of microscopic cellular interaction in post-mortem tissue? Like earlier you mentioned RNA sequencing requires isolating cells, dissolving tissue.
Right, which destroys the spatial context. You know what genes are there, but you don't know where the cells were actually sitting relative to each other. So they used a highly advanced technique called molecular cartography.
But molecular cartography?
Yeah, it allows you to map the exact locations of single RNA transcripts directly within intact human tissue.
Wow, so you preserve the architecture of the battlefield while reading the genetic codes. And what did the map show?
It showed the exact same phenomenon. Right there, clustered around the human amyloid-beta plaques, they found high levels of ISG markers like IRF7 and IFIT1. They found enriched signals for CXCL10, the homing beacon. And they found exhausted T-cells expressing high levels of PD-1 sitting directly in the immediate neighborhood of the plaques.
That is incredible. So the mouse model wasn't an artifact. It perfectly predicted the human pathology.
Exactly.
So recap time for you listening. The arriving T-cells clone themselves to fight the plaques, but the chronic fight causes them to hyperactivate and eventually burn out into a state of cellular exhaustion. And this is proven in both mice and humans. Does this cellular burnout remind you of chronic stress? Like, how long can an immune cell fight an uncleared plaque before it does more harm than good?
Which naturally brings us to the application of all this data.
Yeah.
What does this actually mean for the future of Alzheimer's treatments?
Right.
The most critical takeaway seems to be that the timing of medication is everything. We're looking at a desperate need for time-resolved therapy.
Because the immune landscape is completely flipping. If you have a local microglia-driven response in early disease and then a massive T-cell mediated response in late disease, well, a drug that works beautifully in year 1 might be completely useless or even actively dangerous in year 10.
Exactly. If you administer a drug that suppresses T-cells early on, it might do absolutely nothing because the T-cells haven't infiltrated the brain yet. But if you administer it in the late stages, it could potentially halt that massive damaging neuroinflammation.
So based on the mechanisms we just explored, what would a late-stage drug actually target?
The researchers suggest that specifically targeting that homing beacon, the CXCL10, CXCR3 axis, could be a highly effective way to intervene.
So plugging the nose.
Right. Blocking the CXCR3 receptor, just like they did with the CRISPR knockouts in the lab, could theoretically stop the swarm. Alternatively, you could target the type I interferon response itself. The paper mentions that drugs blocking the interferon receptor, which is known as IFNAR, they're actually already in clinical use.
Really? Like what?
Well, anifrolumab is used for systemic lupus,
Yeah.
and ruxolitinib is FDA approved for other inflammatory disorders.
That's huge. Repurposing existing FDA-approved drugs is always a massive advantage because we already have the safety profiles established. But, there is another real-world application mentioned in this paper that feels incredibly urgent right now. It involves the new anti-amyloid immunotherapies like lecanemab that are constantly making headlines.
Oh, yes. This is perhaps the most immediate clinical implication of this study.
Yeah.
These new therapies use engineered antibodies to target and clear the amyloid-beta plaques from the brain.
Right.
And they have shown success in modestly slowing cognitive decline, but they come with a very serious, very common side effect known as ARIA.
Right, ARA. Amyloid-related imaging abnormalities. It manifests as brain swelling or microbleeds, and it is a major, major risk factor for patients undergoing these new treatments. But how does a swarm of exhausted T-cells connect to brain bleeding caused by a plaque-clearing drug?
Well, think about the peravascular spaces, the areas right around the blood vessels in the brain. As we've established, in late-stage disease, there is a massive presence of T-cells trafficking through these spaces to get to the plaques. You have an army already on site, heavily armed with cytotoxic proteins, but currently in a state of exhaustion.
So they are sleeping, but their weapons are drawn.
Exactly. The hypothesis put forward in the paper is that when you administer high doses of these new anti-amyloid drugs, you are rapidly disaggregating or breaking up the plaques.
Okay.
This sudden, massive change in the local environment combined with the sudden release of plaque fragments, might throw that existing sleeping T-cell army into absolute overdrive.
Oh, wow. So the swelling isn't just a direct chemical reaction to the drug itself. The drug is essentially kicking a hornet's nest. You have a highly localized inflammatory explosion from all these reactivated T cells, and that explosion disturbs the structural integrity of the blood vessel walls,
Leading directly to the edema and bleeding we see as ARIA. It's a highly compelling theory. If the rapid clearance of amyloid exacerbates the background inflammatory response of these T cells, then understanding and potentially calming this specific T cell response before or during immunotherapy, well, that could be the key to preventing ARIA entirely.
That could completely change the safety profile of the most promising Alzheimer's drugs we currently have. It reframes the entire therapeutic approach. We aren't just trying to help the early microglial security guards clear the debris. We have to figure out how to pacify the late-stage T-cell special forces before they burn the building down during the cleanup.
It really is the multi-billion-dollar question in neurology right now. This disease is not a static picture. It is a dynamic, shifting ecosystem.
Okay, so let's do our final recap for the listener. Timing is everything in Alzheimer's treatment. Future therapies might block T-cell migration by targeting CXCR3, and understanding this T-cell army might help us prevent the dangerous side effects of current Alzheimer's drugs. So if you had to target the immune system to cure Alzheimer's, would you focus on the early microglial response or the late T-cell exhaustion phase? It's a lot to consider.
It is. And, there's one final forward-looking detail hidden toward the end of this paper that highlights just how connected this entire ecosystem is.
Oh, what did they find?
The study notes that the APOE4 gene, which is widely known as the most significant genetic risk factor for developing Alzheimer's disease, it has recently been linked to altering peripheral T cells.
Wait, peripheral T cells, like the ones circulating in your body completely outside the brain?
Yes. Your genetics influence how your peripheral T cells behave long before they ever cross the blood-brain barrier. So consider this. If the ultimate damage in late-stage Alzheimer's is driven by this exhausted T cell army, and we know that certain genetics prime those T cells differently, could a simple blood test looking at the baseline activation and exhaustion levels of your peripheral T cells, could that one day predict exactly what kind of immune war will be waged inside your brain?
That is, you could potentially see the tactical makeup of the army years before the invasion even begins. That is a wild thought to end on. It takes everything we thought we knew about an isolated brain disease and connects it to the blood pulsing through our veins. To you listening, keep questioning the systems around you, keep looking for the hidden connections, and keep learning. Thank you so much for joining us on this deep dive.